Thanks. Thanks all for joining. Thank you for participating in this first Webinar Duet. So that's an initiative organized by the CRS-YSC, in which we tried for the first time to pair like a couple of speakers every time, in which they provide different perspectives and their own angle in topics relevant for drug delivery and biomedical technology. And that today is the kickstart of this series that will be composed of a couple of more webinar duets that will be announced in the future. And today before starting, I would like to thank all the people of the team behind the organization of this webinar. So it's not only myself. So my name is Kim, and I'm working at RWTH Aachen University. That's the first one. But with me, there are many other engaged young scientists and people that are supporting this initiative. And I also wanted to thank them. I think it's important. Besides, of course, all the team, we have the executive team of the CRS-YSC with the chair, the immediate past chair, vice chair, and social media lead, together with the headquarters, the CRS headquarters, that have also been supporting our initiative. So thank you to all of you. And without getting more delayed, because I think that people want to listen to the speakers and not to me. So today we have the kickstart of the event and two speakers that are well known, very well known in the field, in the drug delivery field, with years of expertise and experience, not only in the terms of academia, but also we have industry perspective, and that's what we want to bring up today. So the topic of how to transfer scientific ideas from the academia, from the lab, where especially most of us as young scientists are working in the CRS community, which is in PhD, doing PhD or master or postdoc, and working in the lab, and how we can transfer those ideas into industrial development and pharmaceutical pipelines. And for that, we have two experts that will provide that perspective, their experience, and will explain us how to do that or how they have done that, and will give us some do's and don'ts and tips, hopefully, on how to transfer these ideas from the academia to the industry. Just to briefly explain the format, so what we are going to do is basically have two talks first. So Professor Headstorm will start giving his talk. That will be about 20 minutes. Then we will have Dr. Marianne Ashford from AstraZeneca giving also her talk. And at the end, we'll have about 20 to 25 minutes to have an open discussion, question and answer. So what we ask to the people, to the participants in the webinar is place your questions, your answers in the chats or in the Q&A tool. I think there is no Q&A tool, if I'm not wrong. So I think there is. So, but anyhow, you can place the question in the chat, or if you want, raise your hand, and we will allow you to unmute and place the question to our speakers. But please let us organize the people that will ask the question. Do not all of you unmute and bombard the speakers with multiple questions. So without further delay, I would like to introduce Headstorm, and for that we have Amy Logan, who will give the introduction of our speaker. So Amy, please, the floor is yours.
Okay. Thank you, Kim. As Kim mentioned, it's my pleasure to be able to introduce our first speaker, Gert Storm. Gert is an emeritus professor of pharmaceutics and targeted nanomedicine at the Department of Pharmaceutics of the Utrecht University and the Department of Biomaterial Science and Technology of the University of Twente in the Netherlands. He is presently visiting research professor of nanomedicine at the Department of Surgery at the National University of Singapore as well. He obtained his PhD in 1987 at the Utrecht University, and in September 1991, he took up a professor position at the Utrecht University. He is honorary professor of biomolecular drug delivery at the University of Copenhagen and is the founder and chief scientific officer of the company Aovant, co-founder of the company Eden Ophthalmic in Singapore, and vice president of the Phospholipid Research Center in Heidelberg. And he's been coordinating and partnering in many research projects, consortia, and public-private partnerships in the Netherlands and in Europe. He's received several international awards, most recently the Bangham Award at the Liposome Research Days in Glasgow last year, and is active in scientific organisations like ClinAm, CRS, and APV. He's published about 700 publications, and since 2014 is a highly cited researcher listed in the well-known Clarivet list. So without any further ado, over to you, Gert.
Thank you so much for your kind introduction, long introduction. Also, thanks to the organizers for inviting me, and in particular also Quim for organizing this duet and to give a talk in here. This duet with my long-standing friend in science, Marianne Ashford. I thought a duet is always about singing. Do you sing, Marianne? I don't know, but I do, by the way. So maybe at the end of the session we can sing together something. Well, we will.
No, definitely not yet. Definitely not.
No? Okay. Then I do not promise that I sing alone, because if you go from academia to industry, you need to team up. You need to do it together. So I will come back to this in a later slide. So it's actually about this topic, close the gap, concept land, how to come to product land. And you are a scientist in academia, research institute, in a hospital or whatever. You do research and you find something, you have an idea, and you tested it and you found some clinical proof of principle, or not clinical, preclinical proof of principle, and maybe something else, and you think, this would be excellent if it could work in patients. And so how do you do that? So how bring your suitcase with ideas and proof of principle data, how can you bring this to product land? And then, somehow you have to interact with the industry, and how do you do that? And it's all about this in this talk, for the benefit of patients and the market. Well, first question is, of course, why would you want to close the gap? You are in a university, and a professor or a scientist is not a businessman. That's true. So, and to be honest, I think not many of us are willing to go this whole way to bring a product to the market or to the patient. And that is fine. But if you are like that, then you need to have this drive, this personality to really want so, because it's an ambitious pathway. It's not easy, it's not smooth, but it can be very satisfying. And for me, the drive was always, all this work we do in the lab with our formulations. I always asked, would it also work in patients? That was a sort of leading questions. And probably and somewhere in the background, this is dealing with some desire to do something meaningful for ill people. Well, I'm not going to give you a complete set of methods and rules how to do this, but I just go in this short talk through some experience. I had translational experiences and how I dealt with this issue of, let's say, closing the gap. Closing the gap between concept land and product land. And first of all, I would like to say, and I mentioned already, it's very important, if you have this drive, you want this, that you reach out. Really reach out to colleagues, collaborate with them, to other institutes, to clinicians, to industrial scientists, to also sponsors of research. So make your network and collaborate. Team up is very critical, and it's really true. A single twig breaks, but the bundle of twigs is strong, and it's really essential if you go in this direction. Well, you are in the lab, you are in academia, a PhD, a postdoc, or already an associate professor, but you find something really wonderful in your eyes. You have some formulation, and you see some efficacy data, and you have a good idea, and you believe in it, and maybe some safety or something else, and you would like to bring it to product land. Then it's important that you think of patented this first. Take a protection for your idea. IP protection, it is not always needed, but in the far majority of cases, it is essential to become successful. And now, let's start with my first scientific idea in my path with nanomedicine. Actually, it is from a time when the term nanomedicine did not exist yet, and it's from my PhD period. that I did my PhD in the period 1983-1987, and you see here the team, and we were going for a bike tour around Utrecht or something. Yeah, we kept it very simple in these days. And biking in the sunshine, we love it. And this is my supervisor, my promoter, Daan Krommelin, and many of you may know him. And you see me here and some of my colleagues. And Daan Krommelin is already taking the lead. He's really teaming up. You see here, he has the map in his hand and will guide us through the biking tour. And the idea we had at this time, it was a very young group. I was his first PhD student. The idea was to put doxorubicin in liposomes. Doxorubicin, you all know, is a potent anti-tumoral drug, but also very toxic. And can we increase the therapeutic index of doxorubicin by putting it in liposomes? So by targeting the drug to tumors and keep the drug away from those sites in the body which are very sensitive to the toxic actions of the drug, for instance, the heart. So that was the topic. And I think we were very successful. I had 12 chapters in my thesis. And after the ceremony, after the many papers, I was not satisfied with the outcome because I could not prove any tumor targeting with my formulations. And also they were very leaking in the bloodstream. They were going all to the liver and the spleen. I was not happy at all. And that's, I decided to continue, not to give up. And I went to San Francisco. And you see here the well-known bridge, very symbolic for the topic today. And I went for a sabbatical at Liposome Technology in Menlo Park in the Bay Area. And this company, a small company, was developing doxorubicin liposomes for the market, for anti-cancer application. And in that time, it became popular to, it was actually just discovered to coat the liposomes. This is in the 80s, coat the liposomes with polyethylene glycol to give the liposomes a longer circulation time and to diminish uptake by the liver and the spleen, and also to stabilize them against release in the circulation. Now, we did this, and actually, this is the first in-house experiment at LTI, liposome technology, in rats. And you see that we pegylated the liposomes, they were long circulating, and these were the conventional liposomes. So, and of course, this was a long route for the company, but ultimately this led to the product Doxil. And Doxil came on the market in 1995, was approved, the first liposomal anti-cancer drug. Well, during this period, it was a great experience. I learned a lot about how a small company develops a drug product, the way of thinking, the steps to take. So this was really great for me to be in California for some time. The second idea I would like to discuss a little bit, because I have to be fast, 20 minutes are over before you know, is that I came back in the 90s in Utrecht. And the second idea deals with clodronate. You know, clodronate is a biphosphonate, can kill macrophages, but it doesn't enter the macrophages. So you have to put it, you can put it in a nanoparticle, but we did, not me actually, it was Nico van Rooyen, an immunologist in Amsterdam. It is his discovery, his invention. He put it in liposomes, and then the macrophages can phagocytose the liposomes, and then you can kill the macrophages, deplete macrophages. And this is important because macrophages play a key role in many diseases and the progression of diseases, as you know. And we did, together with Nico van Rooyen, we wanted to test if this would work in patients. And we did that in Nijmegen with some rheumatologists in rheumatoid arthritis patients. We gave the liposomes intra-articularly, and so in the inflamed joint. And then we really saw that we have macrophage depletion in the joint, and also relief of pain in that joint, and all the nasty inflammatory symptoms were disappearing. So this was a successful clinical study, a clinical translational study with many clinicians were involved, and particularly Nico van Rooyen, but also you see Nico van Rooyen here. This is him. This is in the 80s. We were in that time in Russia, in St. Petersburg, I remember. And but also Louis van Blois, my technician in the lab, lifelong, for liposome formulations. And he made the clinical batches of clonate liposomes in the hospital pharmacy in Amsterdam, actually. And he did that in a GMP certified environment. And of course, we had approval to do the study, but the aim was, our motivation was not that we wanted to start a company or something, not for industrial development, but we wanted to let patients benefit of something which could work well for them, based on the so-called prescription opportunity that in a hospital, a doctor can prescribe something which is not on the market to a patient, an ill patient, and that can be made in the hospital pharmacy. So prescription services and patients could benefit from that. So that was the basic idea. Later on, after we did this study, there was a company bringing clonate liposomes to the market, but that is because the daughter of Nico Verrooyen, Asje Verrooyen, decided to make a company out of it. And it's now available on the market, but not for patient treatment. It's more for preclinical studies by researchers, a lot of immunology groups in academia, research institutes, and also in companies. They buy, and I think the sales go quite well. Also, Bart Metzelaar, he's now the scientific director of this company, and with him also the company grow towards other applications and other products than clonate liposomes, but other products in food and pharma. Well, subsequent idea I would like to discuss, the third idea I think, is that in the same hospital, Nijmegen, Radboud University, where we did the clonate liposome study, I was also working with the Department of Nuclear Medicine, Peter Laverman, Otto Boelman, Els Dams. And what we were doing is using liposomes, trying to develop them as a radiopharmaceutical to detect sites of infection and sites of inflammation in the patient body. Sometimes you don't know, and you want to know where the inflammation or infection is, and then you can tailor the treatment much better. So this was also a very long project of 10 years. We did many studies, many papers, and we really clinical translated in the hospital. For instance, here you see a patient, and the pegylated liposomes were labeled in this case with isotopes, technetium. You can also use indium or other isotopes, gamma camera image here, and you see that these liposomes, these pegylated, they accumulate in this elbow, but not in this elbow, in this wrist, but not here. This is inflamed. This is not inflamed. This knee is inflamed. This knee is not inflamed. So we were very successful. In this case, it was a rheumatoid arthritis patient. So successful clinical translation, not for industrial development, but based on the same thing to help patients in the hospital on the basis of the prescription opportunity we had and we have. Considering the time, I leave this for what it is. The next example is dealing with Bart Metzelaar again. He was my PhD student in Utrecht. And the idea was, for his thesis, to put corticosteroids in pegylated liposomes to target to sites of inflammation. And in this case, several types of inflammation, but we worked a lot on rheumatoid arthritis. And you see here, for instance, this is one of the animal models, adjuvant arthritis. And you see if you inject technetium liposomes, pegylated liposomes, they really accumulate in all these pores, in the joints of these pores. The targeting is excellent, actually, in these severe inflammation cases. He filled these liposomes with corticosteroids, and we saw a very good therapeutic effects, strong anti-inflammatory effects, sustained also. And of course, the other side of the other reason was also to decrease the nasty side effects of the corticosteroids, and that worked well as well. So we had a strong increase in therapeutic index. Successful. So he founded the company Enceladus Pharmaceuticals in 2005. The funding was based initially on academic grants. Then I had some company awards, then some investors came in, and also pharmaceutical partnerships. And he was able, over the years, and it took a long time, but very successfully, to bring this formulation, Nanocort, to many patients with chronic inflammation, rheumatoid arthritis, as you see many more here, atherosclerotic patients, inflammatory bowel disease, and also actually to cancer patients. And actually, the outcome was also very successful. For rheumatoid arthritis, more than 200 patients, we obtained evidence that it is really working in patients. And this is the frustrating part. We could convincingly show that it works in patients, this product, but there is no product on the market. We are now 2025. We started in 2005. Bart started in 2005. It was a long way. I was in the advisory board, and it took 10, 15 years, and there's no product on the market. We can discuss maybe later about the reasons why this is. Well, next idea, 2 ideas to go, short. We thought maybe we can use these corticosteroid liposomes also to treat ocular inflammation. This was an idea because of my presence often in Singapore, and I worked with some ophthalmologists. They had a PhD student also, and ocular inflammation is everywhere in ophthalmology, and there are many operations, surgeries done, cataracts, retinal, corneal surgeries, and these patients all end up with ocular inflammation. And what you have to do then, as a patient, you come home, you have to use the steroid eye drops, but that is a little bit inconvenient. No, a little bit, it's quite inconvenient. First of all, there's a low bioavailability, less than 5%, but also elderly and kids, and maybe adults also, they don't like to do this all the time, and it's difficult for them. And you have to do this for 4 to 6 weeks, maybe 3 times per day. So it's quite inconvenient, this way to get rid of the ocular inflammation after surgery. So we found, and this is from the clinic, this is a picture from a patient. So this is cataract operation. You take out the old lens, you bring in the new lens, you have another reflex, and this is what the surgeon did. And then at the end of the operation, you give 1 single injection of the corticosteroid liposomes. Subconjunctival. It's just outside the eye, actually. Subconjunctival. And then in the patients we saw, you do not need to use the eye drops anymore. You can completely eliminate the need for using eye drops with a single injection of corticosteroid liposomes at the time of surgery. So that gave enthusiasm to build this company. And we were interested, of course, in many more indications in the eye for the corticosteroid liposomes. And we had the funding, and we started in 2020. And you saw convincing clinical results, but now the company is on hold. Why? Because the investor withdrew. He decided not to continue. And then you're stuck as a company. You have to find new investors, or if it's allowed, what's the contract? So the message here is a little bit, it's better maybe to start with a group of investors, 2, 3, something like this. So don't lean on one if possible. Last, but not least for me, is an idea, very recent one, in immunotherapy of cancer. It's a collaboration with the immunology department in Amsterdam. Again, the same group as of Nico Verrooyen, but so we were doing a project, and the idea was to turn cold tumors into hot tumors. So this is cancer immunotherapy. If you have a lot of active immune cells in the tumor, in the solid tumor, then we call it a hot tumor. If you then give one of the mainstays in the immunotherapy nowadays, checkpoint inhibitors, these antibodies, they will likely respond well, and you see tumor regressions, and you see marvelous effects sometimes. But most of the patients in the clinic, they don't have hot tumors. They have only a low amount of active immune cells there. They have immune cells, but the other immune cells are under control of the tumor. And under control of the tumor cells, they help the tumor to grow, actually, suppressing. But so if you have a cold tumor, and that's often the case in the clinic, then the checkpoint inhibitors will not work, and you have a very low response. So we wanted to convert cold tumors in hot tumors with a liposomal vaccine. It turned out that we ended up with a complete other formulation, not really a vaccine, but with a new lipid nanoparticle containing RNA, which does this very well in animals, in animal tumor models. And here you see one example. This is, for instance, a tumor growing on the flank, and in 10 days, 10 days, the tumor is already at the maximum size. You have to kill the tumor. So it's a very aggressively growing tumor. If you give the immune checkpoint inhibitors alone, you don't see any effect. So this immune checkpoint inhibitors cannot work because likely, and we show that also, it's not likely, it's proven, the tumors are cold. There are not enough active immune cells in the tumor. If you give our formulation, which is changing, we call it RU0134, if you give our nanoparticle, then the composition, the immune cell composition of the tumor is changed. You see already delay in tumor growth. It's already substantial. But if you combine then our treatment with immune checkpoint inhibitors, so we first give this and 1 day later the immune checkpoint inhibitors, you see that the tumor, they melt away. In all the animals, we get cure, we get even a memory effect. So we were very enthusiastic about this, of course. You see another model, this in the liver. If you the liver tumor is growing to large sizes in 2 weeks, if you treat with our formulation, there is no liver tumor growth. And actually, this is a sort of a wish we would like to have, preclinical proof of concept in all these tumors, but we have already in lungs, we have even in pancreas, we are working on breast. So this is our goal, efficacy, a proof of concept for the company. Last year, we had a pre-seed funding from investors. And with Christmas, we were very happy that we could continue with a seed of 2 million. So we have now money to prepare ourselves further, to work with, for instance, the company Lonza for GMP manufacturing. And over half a year, we would like to be in a position that we could go for larger funding, 25 million or so, 20 to 30 million, so-called Series A position, so that we can really go for a clinical trial. And with all the things, the regulatory aspects, the manufacturing aspect, there are so many things you have to cover. And also your preclinical part should be done. But that we are ready to do maybe a clinical study, maybe early 2027. That's our hope. And this is the team. Lucas Sentner is the PhD of that project. He's the founder with me. So we are both the co-founders. And you see, these are very professional people in our team. She's retired from GSK, a medical director, and clinical design is her expertise. This is a business developer. You have clinicians. This is a clinician, Glenn Bonney from Singapore. So a very good team. I have to stop now. The message is, I was speaking about my journey through my translational experiences. Of course, you need to have motivation, drive, hard worker. It's not smooth, but can be very satisfying. But you need to have a network. You need to be social and see people. And you need money. And money is, of course, very essential. Now, I showed examples only based on academic funding. And we did clinical translation of liposomes, and the clodronate example, the radiopharmaceutical example. You can also decide to make your own industry, a spin-off company. Now, Enceladus did this with a mix of academic funding, pharma collaborations, and investors. And Liposoma, Eden Ophthalmic, and now Iovant also is doing it mainly with investments. And then, of course, there's also the option to directly collaborate with the industry, with pharma at an early stage. And maybe the best is yet to come. This is a wink to Marianne. So there is one aspect I would like to mention, not for me only, the clock is ticking with this lecture, but also if you have money, you have to satisfy the milestones of the investor set. You have to work hard. And also, the whole development process cannot take 10, 15 years. You need to be fast in this biotech industry. It's so competitive. You have competing products on the market. And if it's too slow, you may fail. And actually, with this last remark, I would like to give the word to the other singer in this duet. Oh, no, she's not singing. Not talker in this duet, Product Land. Marianne. Thank you.
Thank you, Herb, for the inspiring talk. very, very interesting. So people can already start thinking the questions for you for afterwards. So now I will please ask you to close the presentation and to stop sharing. And I will ask Christina to introduce Marianne. Marianne can already be sharing her presentation. And then Christina, please, the floor is yours.
Thank you so much, Grim. Yeah, I have the great honor to introduce my colleague, Marianne Ashford, who will be presenting next. She is a senior principal scientist, sitting in a global role in advanced drug delivery within pharmaceutical sciences at AstraZeneca. Mariana is responsible for applying drug delivery approaches which enable the progression of innovative medicines, and she's also working to enable novel targets through intracellular delivery of new modalities such as nucleic acid-based drugs. Marianne Ashford has been instrumental in introducing nanomedicines to improve the therapeutic index into the AstraZeneca oncology clinical portfolio. And she has also initiated several collaborations and the building of the internal capabilities in nanomedicines, drug targeting, and intracellular delivery, receiving several internal awards for this work. Marianne has also held project management roles, leading pharmaceutical teams and influencing the global product strategy of various AstraZeneca oncology compounds. She has published over 60 peer-reviewed papers and reviews, 6 book chapters, and holds several patents. Marianne also holds honorary professor roles at the Universities of Nottingham and Manchester in the UK, and is a fellow of the Control Release Society. Here she has served on numerous academic and industrial scientific committees, advisory boards in the field of drug delivery. She's been a long-time supporter of the CRS. She's taken on leadership roles in the UK&I local chapter, as well as being member of the Industry Foresight Council and leading and organizing sessions at several CRS meetings. And those of you who was at the annual meeting last year might remember Marianne, since she got one of our honorary awards at the annual meeting. So Marianne, I'm so happy to welcome you here for this, and we are looking forward to hear your perspective.
Thank you, Christina, for a really nice and long introduction. And thank you, Gert. I knew it would be a very hard act to follow. I've approached this slightly differently. I guess we got the same remit, but slightly differently. But hopefully it's useful. So what I'm going to try and do is through some of the work we've done on really using nanomedicines to enable the next generation of medicines, to try and perhaps highlight collaboration with academia and you know, some of the ideas and some of the translational gaps. It's difficult to do in 20 minutes, so I've tried to pick out a few key areas. And the 2 areas that I'm going to try and focus on, I guess, is nanomedicines to improve therapeutic index and intracellular delivery, and hopefully I'll illustrate why. So I'm just see if I can get a pointer up as well. Okay, so I guess I kind of approached this just over 10 years ago now, where we're kind of looking at you know, reasons for drug failure or why drugs fail during development, and lack of therapeutic index was responsible for about 80% of that. And it's a real challenge when you're discovering medicines and getting things through to patients. So that kind of inspired us to really say that we should be doing more with drug delivery and look to try and use drug delivery approaches to increase clinical proof of concept. So we can use drug delivery as a driver and not just medicinal chemistry, which a lot of big pharma were using to get products to market. And I've really summarized, you know, our first foray into this was to really, you know, use drug delivery to change the efficacy and safety balance, improve that therapeutic window. And we took 2 oncology drugs that had really been were either in development, and one of them was in development, and we hadn't been able to develop them in solid tumors due to lack of therapeutic index, and the other were really failed at GLP tox. And so what we were able to do was take two very different nanoparticle technologies, both in collaboration with small drug delivery companies, and work with them, and then we actually became responsible for both the clinical development and the pharmaceutical development of both of these after the initial preclinical proof of concept, and we took those as far as phase 2. So I'm very pleased and proud that we managed to take two molecular targeted drugs into phase 2 into the clinic, but I guess I'm disappointed we didn't get them further than phase 2. There were very different reasons that I haven't got time to come into why we didn't get further, but really both of them struggled, I guess, in getting that therapeutic index right in very, very different ways. In terms of moving things from academia to industry, just point out they were both with very small companies, Bind Therapeutics, which is really a Bob Langer spin out from MIT, and also sort of Star Pharma, a small Australian company, worked a lot with Monash University to really understand their dendrimer technology. Since then, I guess we've been trying to move as a group to more precision targeted and targeted delivery, and taking that quite difficult journey to really get towards more precision targeting, where I think we need a number of layers of different targeting to make sure that we really are sure that we improve that therapeutic index. And very quickly, I wanted to show you how we're trying to do this in oncology, and credit to the team that I'm working with on this. So we're working with dendrimers, so a polylysine core. We work with sarcosine as our stealth layer, and that provides us prolonged circulation time. It's a generation 5 dendrimer, so we've got 64 functional groups. So half of those have got the sarcosine stealth polymer, if you like, that gives us a prolonged circulation, and half of those we can put drugs onto, so we can carry 32 drug molecules on those, and we use really a hydrolytic linker, and we can also put ligands on that on those. To give us afford that greater degree of precision targeting. And really, right from the very beginning, I guess, this goes to one of the translation elements. These are complex medicines. We need to be able to make them and make them reproducibly. And we've tried to design those for ease of manufacture and scale up, just in terms of the type of polymers that we're using. So very quickly take you through this. So what we did in terms of understanding the platform is put a topo on there, SM38, topoisomerase, so the active drug in irinotecan, and used a HER2 ligand for the HER2 receptor. It's a HER2 Fab, and we can show here that in different cell types we got more. We get greater engagement with in terms of binding and uptake with the cells. We can also show that we can get enhanced delivery to the lysosomes when we put the HER2 ligand on, and we can also see that we get better activity with a ligand over a non-ligand system in vitro. In vivo, in terms of this. When we've got the ligand, by putting the ligand onto the dendrimer, we get a lot more in the tumor. And you can just see in terms of the IVIS on the tumors here, when we've got more ligand on, we get that tumor lighting up more. And on the right hand side, if we look at the average radiance, particularly at the greater time point, we get more delivered to the tumor for when we've got the ligand on and when we've got more ligand on. I think what was also quite interesting, and we found quite interesting, that if we look at the standard dendrimer without the ligand, and this is when we fluorescently labeled ligand, and we have a higher ligand on there. We've got a high density ligand or on there. We see more penetration and more homogeneous distribution within the tumor. And when we've tried to do some image analysis to see what is linked to the stromal compartment and what is linked to the tumor compartment, we can see we've got significantly more within the tumor, particularly at the longer time points. So I guess initially we were quite surprised by this, because we're really adding double the molecular weight onto that dendrimer. And when we did some comparison with an ADC, this is an ADC that we've benchmarked that is doing very well in the clinic, we can show that our targeted dendrimers have very, very similar efficacy in this tumor model to an ADC that has been benchmarked within the clinic. So hopefully what I've tried to show you a very quick story here is that we think we've got nanoparticles here that can complement ADCs. And we know that antibody drug conjugates are now doing very well in the clinic, and we can learn from them. But there are issues. The drug loading is low. There are only about 5 different payloads or types of payloads which are a very potent cytotoxics that have been used. Tolerability in the therapeutic index is still poor. Efficacy is better than the naked drug, but the tolerability is not great, and tissue penetration isn't great. And what we think we can do with our nanoparticles is improve that drug loading, so we can open up the number of payloads and drugs that can be delivered, and we can use multivalency to improve both selectivity and tissue penetration. So that's one example I just wanted to show you quickly. The second area I wanted to just highlight is lipid nanoparticles. And these are really important because they allow us to expand drug target space. Most of the small molecule drugs that we work with interact at the protein level, but if we can interact at siRNA, mRNA level, gene editing, we can really expand that drug target space. We can only prosecute about 30% of the estimated drug targets with both small molecules and antibodies. So if we can deliver nucleic acid, we can really get to far more targets at the moment we can't prosecute. So I don't think with this audience I need to say what lipid nanoparticles are. They've enabled nucleotide-based delivery. They've got those 4 main components. The makeup of those components is critical for the distribution of the drugs. So we debated, I guess, whether we collaborated or whether we brought this technology internally, and we decided to build this technology internally, partly because we felt the LNPs are a really versatile platform. They can be used across different modalities, different nucleotide-based drugs, and a range of applications. I think we're all aware of what they've already done in vaccines. That we're already in phase 3 with Intellia. Kudos to them in phase 3 for therapeutic gene editing for liver diseases. There are early clinical trials where LNPs have been used to deliver for protein replacement. And I'm expecting this year for in vivo CAR T and cell therapies, which should really open up that exciting technology for many more patients, to be in the clinic with LNPs as the delivery system. So we've really set up a design, make, test cycle to try and design the right LNP for the application. And so, both to design the lipids, synthesize the lipids, really try to do some, high throughput in vitro testing to help design and iterate to make sure we get the best LNP as a lead LNP, and then we really work with those LNPs to balance the efficacy, the safety, the stability, and particularly the manufacturability to make sure that we can scale up these, I guess, complex systems and transform them into a drug product. We have invested in some innovative chemistry to build our own lipid library, like many big pharma have done, and this just shows you really how we've perhaps improved with EGFP and improved in expression to the liver as we've learned through our design make test cycle. What we've managed to put in place is some high throughput platforms to support the design of our LNP systems. Both, kudos to colleagues in Sweden who've got this nanoform, which is a high throughput formulation platform, which has allowed us to miniaturize what we're doing, so we can and also efficiently formulate LNPs. And this was from one of our postdoc projects. Michael Munson led the development of a high throughput GAT9 imaging assay that we can use, where we can look at both nanoparticle uptake, endosome escape, and mRNA translation, and split out those different bits to really understand which formulation is best and design our formulations. And in terms of emerging platforms, we're looking at NanoFisher to really look at the high throughput protein corona analysis to allow us to understand more endogenous targeting and distribution. And then NanoScan for more barcoding for a faster in vivo assessment. And I just wanted to show you results of some internal work very briefly, where we've worked with some colleagues in Discovery Sciences that are very good on therapeutic gene editing, and particularly their own Cas9. And this is just one slide to just show where we've used our own LNPs in a disease model of familial hypercholesterolemia, where there's no LDLR receptor, and how we've been able to deliver through targeted cleavage and then an AAV vehicle for insertion of the LDLR, where we've been able to get LDLR receptors and staining and that important gene editing. So just really brief application. So what Kim asked me to do was to try and link some of what we've done and how that perhaps links to our approaches. So I guess our approach, particularly in the early targeting space, and perhaps some of the new modalities that we've worked with, and we've diversified our portfolio in the last few years from small molecules and antibodies to a number of diverse modalities. We've tended to work with smaller companies and companies that have been expert in those modalities, and done some feasibility studies and collaborations with experts. The advantage of doing that is it reduces our risk. There's a higher probability of success. It's a rapid knowledge build, and often there is a development path and line of sight to commercial, but not always. The cons, I guess, of taking that approach is it can be costly. There can be challenges getting agreements, and perhaps a lack of ability to test hypotheses. So it can sometimes be constraining, and there is loss of value because obviously we have to take IP and have royalties and various milestone payments. Often, if we think we want to use a lot of the technology, and as it becomes more mature, we will then internalize, and that allows us to develop the ability to test hypothesis. It gives us greater flexibility to use delivery technologies across different parts of our portfolio. We can actually design, design for delivery and design the compound that fits the technology rather than fit it at the end, which doesn't always work. And it can often be more cost effective. The cons are there are often high technical challenges, freedom to operate, getting IP, and us not having the development capability for that particular technology. I wanted to say something about academic collaborations, and I think we do a lot of academic collaborations, and it's really important that we do collaborate, particularly in difficult areas. We have an AstraZeneca postdoc program. And I think that postdoc program has allowed us to do academic work that has then spun off to greater things. So there are a couple of things I wanted to highlight here. So about 10 years ago, we were asked, well, why weren't we doing more drug delivery in oncology? And one of the things was we don't know which tumor type to develop in and what the difference is cancer is 200 different diseases. Where do we go in terms of development? And I think we were with Jen Moss came in is a postdoc to try and understand nanomedicine accumulation, distribution, and retention in tumors, and understanding which tumor to develop in. And I think we started a very fruitful collaboration with Storm and particularly Tuan Lammers at Aachen, and built an internal team around this to try and understand that and have a number of really nice publications in that space. At the same time, we brought Richard England in as a postdoc in the polymer therapeutics area, and really started to build our knowledge of what polymer therapeutics could do. And that was supported by Cameron at Nottingham, and then had a number of other postdocs in that space. And then we were able to build a small internal team and bring some of these projects into the portfolio. Just on the right here, a number of other collaborations to look at different delivery systems where we perhaps not wanted to invest in building an internal platform, but want to really keep an eye on what's going on and see if we can make sure that we understand and can provide translational support to a number of grants and drug delivery centres. And I just want to highlight Ruth Duncan, I guess, who I think was really inspired that we could do more with drug delivery, designing drug delivery systems to affect products and not just rely on traditional medicinal chemistry. A couple of other areas of collaborating, it really frustrates me that endosome escape is 2%. So we have, or very, very low. And that means that the majority of what we're putting in of some of our most expensive medicines in siRNA and mRNAs, 98% of them get nowhere because they don't even get out the endosome. So we need to be better and have more efficient delivery systems. And I think we've worked with a couple or help set up sort of Formula X and a couple of industry academic pathways and NowCAD with the University of Manchester, and had a couple of really strong collaborations, I think, to try and see if we can really understand what is going on and look at some of the fundamentals of how LNPs work, endosome escape, with a view that we can design better delivery systems. And we've just put out an expert opinion which covers some of the learnings from that, as well as our roadmap to the clinic for mRNA LNPs. So just trying to sort of bring this to the close, I fully believe that delivery will drive the next generation of therapeutics. It's absolutely needed for better targeted delivery. It's absolutely needed for gene therapies and in vivo CAR-T. Traditionally, we have brought delivery in probably close to candidate drug nomination, or people have worked with drugs that have failed in the past. I think we need to, instead of being a passenger here, we need to get back very, very early, and we'll be designed for delivery at TSID. We're not going to have therapeutic gene editing unless we do that, because we're not going to get those projects off the ground. So I think it's more about us really driving from a delivery perspective, and I think that's going to be really important in terms of making sure we've got more successful clinical translation. We also need to make sure we've got a good framework, just as we have for small molecules and other modalities we work for. We need to understand that we've got the right target. We need to show that it can get to the right tissue and the right cell, and we've now got the imaging technologies and other technologies to do that. We've got to make sure we've got the right safety, that we develop in the right patients, that's having the right diagnostics and the right biomarkers, that we've got the right commercial. There are already about 16 or 20 HER2 targeted systems in development. Do we need another one? that we've got the right culture, because this is doing things differently, and that we've got the right developability, that we can actually make and scale up and characterize these complex medicines. I've used this slide and I just put it in at the end. Previously, we are quite good at treating rodents. We're not so good at treating patients, or not so good at treating a huge patient populations. I think it's relatively easy to make some of these systems in the lab at pilot scale and kilogram scales, but can we do it efficiently at hundreds of kilogram scales if we need it? You could argue if we're really targeting, that will reduce the volumes that we need, and that will be more efficient and more sustainable. We need to ensure that we've got the quality by design, and we need to ensure that we've got the right methods in place to make sure we can assure the quality of these products. Collaboration is absolutely key. A medicine is more than a molecule. We need to get drug delivery more in the design phase, and we need to get all the relevant skills and really work well supportively together, both across disciplines and across sectors. And before I finish, I just want to acknowledge my many colleagues in ADD who have worked on some of the modalities I've shared and some of the tests I've shared across all our sites, and particularly highlight Richard England and team and the polymer work that has allowed us to really push things into the portfolio. Colleagues across the different functions in AZ and many of our external collaborators. So thank you, and thank you for your time.
Thank you so much, Marianne, and thank you so much, Gert, for this really rich talks. Thank you for time and all the interesting talks. Let me please start the Q&A session, and feel free, both of you, to be involved in the answers as you like. My first question from Ruth. Thanks for sharing all your personal experience. You mentioned various challenges to bridge the gap. What would you say is the #1 challenge for academics bringing their idea to the patient market? Is it money, manufacturing, scale-up, regulations, or finding clinical indications where their idea can make the difference, or anything else? So please feel free to answer.
Yeah, that is a question for me. Yeah, all these aspects mentioned is really critical to get it flowing and to be successful. I think if you are a scientist in academia, in a university, and you want to go in this direction, I think there are two major steps. Not just, maybe I could say #1 is the money, but before that, there is also the right spirit, your experience, to build a good team. You need a team with people who understand what the pathway to product land is. And that is all these translational topics. It's manufacturing, of course, scale up, regulatory, and also clinical indications. I think it's best if you have a sort of disruptive indication, so that you really make a difference in the clinic, because ultimately you need to convince investors. And then I come to the money. It's not easy to get money, and if we focus on investors, then they have their demands. It's not easy to get. You need to really differentiate yourself from others, because, and I think in Holland, I don't know, they have so many spin-offs. Most of them fail. They get some VC funding, but most of them have failed in Holland. And so the risk of failure is high. So getting the money in Europe, it's anyway also not so easy to get enough money to do what you need to do, because investors are less risk taking than, for instance, in America, where there is much more money. They also are much more inspired by your idea, how important it is, and so they are willing to take higher risks. So actually, we moved the company already to America, and you see it helps, because our funding is not from Europe, but from another part in the world, and South America in this case. So it is this story that you need to find the money, and you need to train and to build a team. I think these are the most critical steps. So that means you have to go out, train yourself, build a team. It takes time. And you cannot do it after the experiments and you see, oh, this is nicely working. Oh, okay, I want to build a company. No, it doesn't work. You need some start money. So mostly it's low money from the government, the local government in the Netherlands or whatever. You can do something more, but you have to build your team. And all the expertise needed need to be in the team. So this is my answer to this question, I think.
Thank you. We have another question from Thes. From an academic perspective, I'd like to ask this. This is also open for Gert and Marianne. Many times we have a project company which is able to supply an end-to-end solution to take your RNA therapeutic or other idea through preclinical testing with their nanoparticle formulation. However, then will it charge many thousands of euros? Thus, those in academia are more convinced to synthesize and formulate everything ourselves. Do you have any thoughts on which might be preferable?
Maybe Marianne first now. First.
Thank you. Yeah. Okay, so I think if I understand the question, is it becomes too expensive to collaborate with people, but please say if I've got, because they're charging a lot of euros, so therefore there's a bit of a driver for doing it yourself. I think that's the same in industry.
Yeah,
you know,
that's
it's one
great.
of the reasons
Yeah.
why we've gone down. Is that have I understood the question correctly?
Yeah, you have that basically it's too expensive, and also in many cases they would end up keeping the IP and having a say in how you progress with that product.
No, I think that's why if you look at LNPs, a lot of people and a lot of big pharma have also making their own lipids and started the LNPs. And I did have a slide in, should we buy or should we build? I think we thought the platform was broad enough, there are enough mRNA guides, therapeutic gene editing coming through, that it would be a benefit us to build. But using it costs to build because we have to bring people in, build capability, but equally it's very expensive to do it externally. So it's a real balance to try and work through. And often we kind of start collaborating, and then if we think it could be used across the portfolio, you would build internally. So I guess that's the same if you want to do it in academia. I guess the question is, have you got all the controls that you need if you're trying to get into the clinic? How far can you realistically take it? And if you're an academic, do you want to do that? Or is it, it depends on whether it's a different career, I guess, if you get something and you then go and spin it out or build it, than it is the career paths you started in academia. Some people like the flexibility of lots of new ideas coming through and sending them off in different directions. Some people want to translate. So I guess it depends.
And maybe I can add
makes
to
a lot of
it.
sense.
And maybe I can add to it, if you are already in big pharma or something, the money aspect is important because internally you need to create much more money. But if you are in academia, that's a different story with money, of course. We all feel that. And so we have used quite a lot also contract research organizations for doing specific experiments, for instance, in a certain tumor model which is running in that contract. And we then got the results, and we are very critical on those contract research organizations for doing work if they are accepted by the FDA, for instance, if they are known by the FDA or by the EMA, so that they have a reputation in this field. So, but you need money to do that. It's costly. So you need first, money is the biggest obstacle.
Yeah, indeed. Thanks for the answers.
Thank you so much. I believe Marianne also have a question. Could you please go on? Mariana. Maria? Hi.
Hi.
Yes.
So now we have also a question from the chat. I can ask mine in the final. We have a question from Jen Nicholas May. Which idea that did not turn into a product broke your heart the most? Could be due to the idea, application, or teeny details missing to make it on the market? I think it's for both speakers.
Well, Marianne, please sing.
You don't give me any thinking time.
Oh, that's the reason why I give you the word first.
Thank you. At least we're not singing together. Okay. I guess, Jan Nicholas, thank you for the question. I guess my slight frustration, I hope I don't get into trouble for this, is that it was great to be able to put nanomedicine projects into the portfolio. We needed a lot of capability build, and it was difficult to persuade people to take a slightly different path. So we tend to treat them like a typical small molecule and do a standard phase 1. So we didn't do biodistribution early. We struggled to get some patient selection in, etc. And part of that, it goes down to costs. They're more expensive than a standard formulation. So asking for extra studies and extra money to get that product understanding was a challenge. And I guess that's a frustration. I try not to let my heart get too broken for too long.
Well, and I could add, my frustration is that this work we put in the corticosteroid liposomes, which appear to work in the patients, they do what the idea was, and they are therapeutically useful, are not entering the market. So is not becoming available for patient treatment. Breaking the heart, yeah, I would say it's really frustrating. And maybe that's why with this new company, Iovant, I'm learning my lesson. You know, you are inexperienced. I was inexperienced in academia, and you do what you think. There was money from organizations, also for valorization in Holland is the name, for clinical translation, translational studies. So you take that money, that's academia, you write projects, you get the money, you start, but everything is slow. You have a project of 4 years, you have money, you make some steps, and then you need another project, and so that went very slow. And I think this whole development of corticosteroid liposomes for the treatment of chronic inflammation, in particular rheumatoid arthritis, was too slow. Started in 2005, and it we are now 2025, but yeah, already 5 years we know it's not working. It will not become a product on the market because that is so competitive. Other products came on the market. The whole market changed. There are now antibodies against TNF alpha, infliximab, and all this. And that makes also the interest of your exit partners, pharma. At some point, you have to give it to pharma for further development, but they need to convince that they have a good market niche and that they can really make their profits from there. And that changed a lot. And it's easy to talk from hindsight. We didn't know, and I've learned. So it's not a critical comment. It went like it is, but it's frustrating. And yeah, I hope that we can now take this as a lesson. And with my current company, Iovant, we can do it better and really be fast and move to the clinic as fast as possible in a responsible way, of course.
We have other question from the audience that this time from Amy Logan. You both have a lot of experience working or collaborating in many countries. In your experience, is there any particular country that are better at connecting academy or industry, and any reasons why?
Shall I answer?
Yeah. You can start.
Yeah. Okay, because I think not really. I think it depends on your on your personalities, on how you connect to people, how you bridge with people, how good are you in selling your idea and convince other people. I think that's more important than a country in which academia and industry is not well connected. Actually, in Holland, we don't have any bigger pharma left. So we have to look for other countries. But I think the world is open and wide. With the corticosteroids, liposomals, it was an Indian company, Sun Pharma, was very much interested in and paid all the clinical trials. So I think it depends on your ability to connect with others and to convince them. Maybe Mariana, how do you feel?
I think I would agree with that. I think it very much depends on individuals, and I'd probably say it's more to do with that than particular countries. In my experience, I've had, you can say I've had good collaborations and not so good collaborations, and it's down to the individual and the approach to work often, and how well perhaps the chemistry works between the collaborators.
Okay. I also have a question. In your opinion, what is failing in the conceptualization of the projects from the academia to do the well translation into the industry? Also, you mentioned that the translation should be fast. How fast should be, since we need to have a robust device, medical device, in that case in nanomedicine and nano device, to be implemented later on in the clinic. You mentioned that you have a lack of animal experimentals. We have to do a throughput screening of the strategies. But what should be the biggest reason how projects are failing the translation?
Probably it's a question to me because I said fast. Maybe I should have added the word fast enough.
Yes.
Of course, you cannot do it next week. It takes time. It's a complex job. It's ambitious. And there are many, many aspects you need to cover. And you can fail along the way, and it takes longer, or you have to reformulate maybe, and all takes time. But it should not be so much time that other people working in the same field come with competing products on the market, for instance. And they have a share of the market, and they do maybe similar things or whatever. So if you wait too long in the biotech industry, I think you will fail. It cannot take 10, 15 years. We hope, for instance, in 2028 or something, that we have some clinical data. This is key, of course, to have some data from patients showing that your ideas work. If you have that and you are in an excellent niche of the market, the drug market, which is attractive for pharma, then you are in a very good shape. So getting your clinical proof of principle, that's key. And that should not take 10 years. That should be as fast as possible. But I cannot give exactly a second more or a second less for this. But I try to convey the feeling with it. So also it means don't rely too much only on academic grants, because that makes small steps and it takes time. Maybe you have a patent, also your patent expires. So in time, because you have to protect, but in time it can expire. So this is all in the game. And that also makes it, very challenging and complex, but also, the feeling that you work on something really meaningful is very satisfying to me, at least.
And I think just to add to that, if you're working on a competitive target, it's liable that your standard of care will change. So you've done a clinical trial with the wrong comparator. So if you take too long, the longer you take over it, the more likely that is to happen.
Okay, thank you. Now we are going to move for our colleague Leila Rabi. I know that she has also some questions, and she will in charge for the questions from the chat.
Thank you, Maria, and hello everybody, and thanks for the great talk Marianne and Kurt. So I will continue the question from chat box, and then I will ask my own question at the end. So I see the question, it's for Kurt, I think. Actually, it's the same concept. How did your past failures impact your ability to secure funding from investors? And additionally, how do you view Asian countries? I think he means Singapore probably as potential alternatives to the US to for fostering startups. Please.
Yeah, I think I already partly answered this with the remark you need to be fast, and I had failures, and but about Asian countries, yeah, then we of course think of China and Singapore, and actually I investigated Singapore, but the biotech activity in Singapore is not so huge. It's actually very small. But in China, it is of course very active, and that is of course a potential alternative also to go Asia for your funding. And but everything depends on the conditions. Depends on what is the future. Can you make contracts which are agreeable to all the parties? That is the key, the bottom line of all these negotiations, of course. And China is a big market. America is a big market. Marianne knows better than I do, but you should not exclude Asia, of course. No, not at all.
Or maybe could we have your perspective, Marianne?
I think it's important to make sure that we look globally for collaborations, and I think the pace of change in China is particularly rapid. I think for things like cell therapies, I think it's easier for to do clinical trials in China because they've got an IIT intention to treat protocols, so that so certainly in areas like that, I think the innovation is quite different. I think I heard this morning that at JP Morgan, there's a lot more. 5 years ago, it was global-led innovation and people trying to develop in China for China. But now that the innovation is leading on the China side, so it's innovation and development. That's just one Asian country, but I really think that innovation is globally, and we need to collaborate globally.
And the next question is for you, Marianne. It's about your experience at AstraZeneca. How the knowledge you got internally in this company can be shared with academia so people won't repeat the same mistakes.
So I guess we are trying to publish, and we are pushed and encouraged to publish, and we both publish and accept presentations and talks to try and share that. So we are trying, I guess, hard to share both successes and programs that perhaps have not gone as we've wished. We do put also quite a lot on our website in terms of what our strategy is, and we kind of update what we're doing in drug delivery in other areas every couple of years. And we also try and make some of our compounds that perhaps haven't been successful open for others to do collaborative work in an open innovation type model. So I think it's, when I started in the industry, we didn't share anything. We weren't encouraged to publish. And everything had to be held close. But now I think it's a very different environment where we are trying to share and collaborate really to make sure that we can accelerate medicines development, for patients, and we can all learn from that. Separately, I try and do a little bit of translation and input to both companies and grants to try and, academic grants to try and help progress novel ideas and give the benefit of our learnings to others. So I think in a number of ways, and I know a lot of my colleagues do that as well. We have a lot of colleagues out presenting at conferences and posters to try and share our work. So we do try. It's probably not perfect. Also happy to be, approached and share what I can.
Great, thank you. And I will continue with the questions from audience, and I will put my question at the end if we have time. So, Tara asked about the other nanoparticles like exosomes. Could you please say, do you see any future for exosomes in clinical for treatment of different diseases? For both, I think.
Are you going to put me first again yet?
Yeah,
Because I don't think about this answer. No, it's you first.
So, I mean, there are exosomes in the clinic. I think they're early in the clinic. Also, people are trying to use exosomes for diagnostics. I think they might get there for early detection sooner than they perhaps will as therapeutics. I think some of the challenges with exosomes is their characterization and be able to make them in high enough numbers. And also, I think we really need to understand exactly how they work.
I cannot add more.
I've truly, and I've got a colleague. I don't know if she's sorry yet.
Oh, I say you said it very nicely. It's my opinion also for certain purposes, like for diagnosis, it's really attractive, but they are complex systems and not characterized very well. And you have so many papers and so many claims. I still have the feeling that overclaiming in this field is playing quite a role. I don't say it's impossible, but exosomes for targeted drug delivery, for instance, I think is still quite a way to go. I do not want to discourage people, but they are biological systems which are difficult to characterize. So in general, I always feel if you would like to make business or go and make a spin-off company, keep it simple. And the less elements you have in your product, the better it is, because every element needs to be characterized, and you're maybe dependent on another supplier. And in the case of exosomes, you don't know exactly what's all in there, and probably will be special regulatory requirements for these systems. But for me, it's not a simple system yet, and that's why people also trying to make it more simple, and this should be encouraged. So, but for now, I think please go on. We are not there yet with the exosome.
Regarding the simplicity, I would ask my question here because it's related to your talk. And what's your idea about the functionalized nanoparticle, ligand functionalized nanoparticles? What's the future in the clinic?
Maybe Marianne has one in the clinic. She discussed it in her talk. So, Marianne?
No, it's not. Unfortunately, it's not in the clinic. But I guess ADCs are functionalized nanocarriers. I know that's controversial for those in the ADC world, but I would say they're biological nanocarriers and they're functionalized. I think they have shown the benefits of targeting. I think some of the radioimmunoconjugates have shown the benefits of targeting. You need a lot less, if you target, you need a lot less drug. I think targeting is here to stay. I agree they're complex. There's a lot more we need to understand, particularly about receptor recycling rates, resynthesis rates, intracellular drug release, etc., and whether it needs to be released. We could cite really well-designed ADCs and poor-designed ADCs and what they do in terms of benefits to patients. But I do feel that we will have targeted nanoparticles. We've got antisense oligonucleotides with GalNAcs on them, reducing the dose tenfold. We just need to understand the rules better. I don't think we're quite there, but I think we've also got a lot of imaging tools and other tools that will help us understand more. So we've got to go there.
But if you don't need
maybe
the target,
sorry, 1 second. Just to announce to everyone, so we're in the last 5 minutes. I know there are many questions, so if Marianne heard, just for you to know, if we can stay 5 minutes more. I know that I said that it's going to be about 1 hour, 20 minutes, but it's appreciated. So we will have the last couple of questions. And for the audience, if many or more people have questions, please put them in the chat and we will find a way to hopefully reach out to Hert and Marianne, and maybe they can either answer in by email or something. So that's all, so everyone knows that. Sorry, Hert, to interrupt you. Go ahead, please.
You give the word to me, or?
Yes, Hurt, go ahead. I just wanted to
It
answer.
was about the targeting ligand. As long the target ligand is really adding something. What I read too much in the literature, targeting ligands we can better target. That is not often the case. That's not always the case. It's at least for the localization in your target tissue, the targeting ligand is more a barrier sometimes because you have other pharmacokinetics or whatever than helpful. But a targeting ligand might be needed to get cellular internalization by your target cells. And if your aim is that, it's justified because if it without you are not getting into the cells, you need it. But so I agree that this will lead to something, but you need to realize the function of your targeting ligand. And it is not a molecule which is sniffing around in the body and binding to your disease site.
But also, you may not need it. There may be drugs where you just need a slight improvement in therapeutic index, and you just might be able to do that with control release, accumulation in tumors, a bit of molecular targeting. So don't do more than you need to do. Don't over-engineer the delivery system because that adds complexity, adds cost, and probably increases the probability of failure.
Thank you so much. So we don't have lots of time, so I asked Eva. I think I saw you raise your hand, so if you could ask your question.
Yes, I have just a final question. Like, how a young scientist would evaluate his idea? Is it suitable for industry without losing its novelty? Keeping in mind that he takes all your advice and all these steps in mind, but still in our research, we need like an external eye, So how can we do this without losing the novelty?
I think you need to be quite hard on yourself. And if you look at, put together a target product profile and say, really what do I need to achieve where? Or look at, we do the 5 Rs, Pfizer do something else, have I got the right target, right exposure, and really assess your idea versus those in a really hard, objective way. I've done things like, a Kepner-Trago analysis to say, what does this need? And therefore we can rank order delivery systems or rank order where you are. Because sometimes it's very hard if this is your pet project, it's hard to be objective. And I think you really need to push yourself to be objective. And then you can find the gaps and work out whether you can address those gaps or not. So I think there's enough written, and there's enough papers now that have been published on translation and what you need, and people have recognized the translational gap. So I would take a couple of those, matrices or whatever, and really be really hard on yourself. Because I think by doing that, you'll improve your idea, you'll refine it, and you'll be able to address those gaps. I don't know if that helps.
Yeah, of course it helps.
And you can use consultants or whatever, or supervisors, or get somebody else to really challenge your thinking.
Yeah. Thank you so much, Marianne. Thank you so much, Gert. And Kim, would you like to with a final conclusion?
So maybe I will take over. So there is still one last question. Maybe Marianne and her can take 1 minute to answer. So Jan Nicholas also has a second question. And he's asking how important is, on the one hand, getting clinical data in a fast way, but on the other hand, ensuring that the right patients who are likely to respond to your therapy are enrolled in your clinical trial, as non-convincing data may also harm your long-term success, and there might only be a limited number of attempts. I guess that's a question that maybe Marianne can start, I guess, for applies to the biomarkers paper. So I think that we can start.
Okay, so we do a 5R or 6R. One of them is right patient. If you don't go to the right patient, you're going to get the wrong results. And we need to make sure that we've got the right diagnostic there, or you've got something to make sure you get to the right patients. I think you could look at a number of clinical trials that have been done and carried out poorly, and I can name a number of technologies that have not mated, and it's tainted the whole technology by doing the wrong clinical trial at the beginning. So that's my take on that, young Nicholas.
Yeah, this is a sort of proof of the pudding question also for the company which we started. So what is the right patient? What is the right indication? And of course, if you are not getting positive clinical data in your first clinical trial, it's very frustrating, and it's maybe also end of story already. Maybe not. That depends. So to be honest, we follow because we have a sort of, nowadays you have a lot of personalized therapies. It's CAR T, it's for that specific, it's from the patient, and actually it's very laborious, and you take cells from the patient. That sort of, it's then you know what the right patient is. But in our case, we have a sort of more generally applicable nanoparticle system for treating tumors. And there is a big variety of tumor patients, of course. And so in our clinical design, we decided to go for a sort of basket design in which you can include more types of patients. And so we have a sort of screening which tumor types would be sensitive to our treatment and which not. And then we do this already during our safety phase 1 study, which we plan. So this is all being set up, but it's a very good question because, finding efficacy data and while the formulation is safe is, of course, the key to proceed.
Thank you. I think that's a very nice and message for the webinar. I would like to thank you both again for accepting the invitation, for being part of this kickoff, the event of the Webinar Duet series. It was a pleasure to have you both. Thank you for the talks and for the inspiring insight. And to close the session, I just would like to remind the audience that we already have organized a second Webinar Duet. It's going to take place on Wednesday, 5th of March at 9 a.m. Eastern Time and 3 p.m. Central European Time. And the two speakers will be experts on how to implement artificial intelligence on the pharmaceutical industry or on formulation procedures and also on clinical practice. So please stay, sorry, I missed one slide. Yeah, stay connected to our social media, Twitter, LinkedIn, Facebook, or even contact us via email to the CRS Young Scientist Committee. Thank you all for joining. Thank you again, the speaker, very much. And have a good evening or a good morning, depending on the time zone. Thank you very much. Take care.
Yeah,
bye-bye.
Thank you for the invitation. Bye-bye.
Thank you, Marianne. Thank you all. Hope to see you once in the future.
Thank you. Bye.
Bye-bye.
Thank you.
Bye.